Hi everyone, today I'm delighted to have the CEO of Amplia Therapeutics, Chris Burns, on the podcast to talk about the company and the exciting developments and clinical results they've had recently. So Chris, welcome.
Hi, Michael. Glad to be here.
So I thought it'd be interesting to just take a step back and talk about the disease that the primary focus of the company and the disease that you're treating. So you're able to talk about pancreatic cancer, how the disease appears, how it's diagnosed, and perhaps we can move on to some of the difficulties that clinicians have had in treating it.
Yeah, certainly. So, pancreatic cancer is a cancer of the pancreas, which is an organ that sits deep in the body below the stomach. And the cancer is often quite difficult to diagnose because it's buried deep in your body. Patients often don't realize there's disease associated with it until quite late in the disease process, which means that over 50% of patients are diagnosed with metastatic disease, meaning disease that's already spread to other parts of the body, which makes it, at that point, very difficult to treat. Metastatic pancreatic cancer has a 5-year survival of 13%, which is one of the lowest survivals of any cancer. So, in other words, if you're diagnosed with with pancreatic cancer, I should say, today, a 13% chance you'll be alive in 5 years. If you're diagnosed with metastatic disease, so that's the more advanced form, you've got about a 3% chance of being alive in 5 years. So, very challenging disease to treat, and clearly patients need better options and need better outcomes. So our focus is very much on targeting pancreatic cancer and the metastatic disease in particular. As we talk today, we'll talk through our approach and where we're getting to.
Yeah, absolutely. So how do patients present? What are the key symptoms they'll have? And is surgery is usually the first option if it's possible. Is that
Correct.
correct?
Yeah. So patients will often present with an array of different symptoms, actually. It can be as mild as say, back pain or altered bowel habit. It can be jaundice. It can indeed even be diagnosed after someone's been diagnosed with type 1 diabetes. So if you aren't diagnosed with diabetes, it's always good to check that you don't have a problem with your pancreas. So there's a range of reasons that people will be diagnosed, but often they're kind of mild things, stomach pain, maybe altered bowel habit that people will put down to something else, which often means, of course, it's diagnosed quite late. So I think that's one of the real challenges with the disease. And unfortunately, what we're seeing is that around the world, pancreatic cancer is predicted to be by 2030 the second biggest cancer killer. So that's obviously really problematic. That's partly because some cancers are getting better treated, but also because there is an increasing incidence. Unfortunately, that's across the board in terms of ages, and indeed the biggest increase in incidence is younger patients over 30 and above. Important that if you do have any of those symptoms that you actually check out. That's not a problem with pancreatic cancer.
And so what are the treatment? So you go into clinic, you've been diagnosed. What are the treatment options at that stage?
So it depends whether it's metastatic or not. If it's not metastatic, you mentioned surgery, that's often the best treatment. So if it's only a cancer growing on the pancreas itself, that can be surgically removed if it's not too developed on the pancreas. It's obviously a difficult organ to get into the body just because of its location. So that can be challenging surgery. So there are treatments for some chemotherapy treatments for local disease, but sometimes that local disease, when it's just on the pancreas, is too large for surgical removal. So there's treatment. And Oncosil, for instance, on the ASX are working in this space of treating what's called non-resectable locally advanced disease, where it can't be surgically removed at the point of presentation, but there's a therapeutic treatment and you shrink the tumour so that you can remove it surgically. So locally advanced disease is one stepping stone between something which can be treated with therapy, with surgery, and metastatic disease, which is obviously very difficult to treat and treated largely by chemotherapy. And in the middle, you've got this locally advanced. Radiation can also be used for disease that's just in the pancreas. If we're talking about metastatic disease, where it's gone to elsewhere in the body, the main treatments, probably over 75% of patients are treated with one of two types of chemotherapy. The first type's called gemcitabine and Abraxane, so it's two drugs that are given on a cycle of 3 doses every 4 weeks. That chemotherapy is reasonably well tolerated. It does have some long-term toxicity. And for patients who are getting gemcitabine Abraxane, you're looking that there'll be an extension of survival, maybe up to sort of 8 to 9 months. But, it's not a great outcome. The other main therapy is called FOLFIRINOX, which is a complex name. It's actually 4 drugs that are given on a 2-weekly cycle, so every 2 weeks. That's a much more aggressive treatment. It's not as well tolerated by patients from the outset. Typically, patients who are on FOLFIRINOX will see a slightly improved overall survival of around 11 months. But even, as I said, the 5-year survival for metastatic pancreatic cancer is very low. And unfortunately, with either of these 2 treatments, gemcitabine Abraxane or FOLFIRINOX, the median overall survival is less than 12 months.
Right. And I understand, FOLFIRINOX is more popular in the United States. In Australia, perhaps you're more likely to get the other version. Presumably that's due to the intensity of the treatment. How serious are these side effects? What are we talking about here?
So with gemcitabine Abraxane, the longer-term side effects are some cytopenias, so effects on your blood counts, decrease in certain cells in your blood. That's from the gemcitabine. The Abraxane can cause something that's really quite debilitating, and that's peripheral neuropathy, where basically you lose feeling in your fingers and your toes, so it becomes very hard to eat or to button your clothes, those kind of things. So we've had a couple of patients on our trials who've actually come off study because of either the cytopenias or the peripheral neuropathy caused by the chemotherapy. So they're not great, but initially and for definitely 6 months, patients generally do reasonably well on those chemotherapies. FOLFIRINOX is more aggressive, so it's generally kept for younger, fitter patients. So, as you say, in America, they have a bit more of a heroic approach in treating cancer, so they really go very hard. But the side effects are the sort of more classical side effects we think of when we think of chemotherapy, so hair falling out, a lot of gastrointestinal upset, vomiting, diarrhea, those kind of things. And unfortunately, that happens really from the first dose onwards. So it's a very challenging treatment, and obviously any way that we can to modify the aggressiveness of those treatments, maybe dial back some of the toxicity of some of the dose, so we dial back some of the toxicity, will be, I think, really well met by patients and their families. That's a longer-term goal of ours.
Yeah, it's a difficult trade-off.
Yeah.
Yeah, difficult trade-off for the extra couple of months. And so why has pancreatic cancer been so difficult to treat? Why are the survival rates not improved like they have in other solid tumours?
Yeah, well, it's a good question. One of the reasons is that pancreatic cancer itself is what we call a fibrotic tumour. So it's if you actually look at the tumour tissue itself, it's very hard. It's got this sort of scar tissue, this fibrous tissue that sits around it, so that it acts as a barrier essentially for any therapies that we might give patients, or indeed even your own immune cells can't really penetrate and get into the tumour and actually attack it. So you've got this fibrous surrounding tissue. That's one major problem. That leads to reduced immune attack. It leads to a difficulty for treating with chemotherapy. So that's one reason. I think the other reason is that cancer cells themselves have a number of different genetic mutations that lead to an aggressive type of cancer. So they grow quite aggressively, these cancers. And I've heard stories of people having the cancer detected by the time they go into surgery, which might be a couple of weeks, maybe a month later, the tumour's grown substantially in that time frame. So you do have a lot of different genetic drivers that are driving the tumour cells themselves to make it, again, quite difficult to treat.
So that going to that first point, that stroma, that fibrotic shell, is that one of the reasons why drugs have difficulty penetrating and being as effective as they are in other solid tumours?
Yeah, exactly. So fibrosis is a response that's kind of brought about by the cells that sit around the cancer cells themselves. And it's a response really to inflammation. So there's this sort of inflammatory component that leads then to the fibrous tissue, and that really does block drug penetration and does block cell penetration, even biological species like antibodies really struggle to get into that tissue. And that's a real challenge. And our drug is designed to inhibit a protein that contributes to that fibrotic process. Our drug, at least one of the ways it works, is to break down that fibrous tissue somewhat. It doesn't break it down completely, but it relaxes it to the point where you do get better penetration of drugs, better penetration of immune cells into the tumour tissue, and so that you can attack the tumour cells themselves.
I guess that's a good point to talk about FAK inhibitors in your drug. So you mentioned one way it works. So what is the therapeutic hypothesis? How do you think it's actually working, or what's the mechanism by which you're getting your results?
So there's a couple of different mechanisms at play. So the first is this effect on the tumour microenvironment, where we're modulating the fibrosis, modulating this fibrous tissue and allowing better attack by drugs. You're also allowing better attack by the immune cells as well. So your body can mount an immune attack against the cancer cells, but it's difficult with that fibrous shield in place. So there's that effect on the microenvironment, and there's a second effect where the drugs actually target the cancer cells themselves. So our drug inhibits a particular protein that is overexpressed in pancreatic cancer that contributes to cell survival, that contributes to cell migration and proliferation. So by inhibiting the FAK enzyme, we're blocking those processes that are happening actually in the cancer cell. And we're also blocking resistance pathways that might develop over time, say in the presence of a chemotherapy or indeed a targeted therapy. FAK activity is often upregulated in response to those other drugs, and so by blocking FAK, we're blocking those chemoresistance pathways. So we like to say it's a 1-2 punch. There's the punch on the tumour microenvironment, making that much more impenetrable by drugs, and the second punch on the cancer cells themselves.
Have FAK inhibitors been tried before, and what was the outcome?
Yeah, so FAK and FAK inhibitors have been around for, what are we now, probably getting on to 20 years. The very first generation compounds weren't particularly selective or potent, and they had a lot of other issues. All the preclinical data, so data in mouse models and other models, indicated that FAK inhibitors would work in pancreatic cancer, and so a couple were taken into clinical studies, but because those drugs weren't particularly optimized, those early trials didn't work. We became really interested in progressing our particular molecule because we started the company Amplia here in Melbourne to commercialise a drug that was discovered at a industry academic partnership in Australia called the Cancer Therapeutics Cooperative Research Centre. So this was a team of academics and industry people that came together around specific problems in drug discovery, and one of them was developing inhibitors of FAK. So we were aware of this drug. It was a very good drug in terms of the preclinical data that the team had developed, but it wasn't being taken into the clinic. So we started Amplia to develop this Australian drug in cancer. And we became interested in sort of 2015. There were some papers coming out of the scientific literature showing that FAK inhibitors were very much involved in modulating the tumour microenvironment. That hadn't really been appreciated up to that point. And knowing, of course, that pancreatic cancer has this fibrous shield, if you like, around the cancer, we thought moving the drug forward into the clinic with a particular focus in pancreatic cancer was a sensible thing to do. And we teamed up actually with a group of scientists, a very talented group at the Garvan Institute in Sydney, who have a lot of expertise in pancreatic cancer, and particularly this fibrous tumour microenvironment. So that was Paul Timpson, his team, Professor Paul Timpson. So we worked with Paul and really showed that our drug actually did what we thought it should do in various models of pancreatic cancer. And so it's really with that data that we started the clinical program. I think that answered your question, Michael. I'm not sure exactly if I went off.
Yeah, that's helpful.
Off piece, but yeah, that's how we started.
Yeah, and it's helpful to understand the background to FAK inhibition and potentially why other previous approaches didn't work so well. Why don't we move on to your accent trial, and are you able to just run through the results now that we have them?
Certainly.
And what you're expecting, what was like the threshold for success, and yeah, how the trial was designed and ultimately the result.
Yeah, absolutely. So the ACCENT trial is a trial we're still running. It's a trial in metastatic pancreatic cancer. We give our drug that we now call namafotinib. It was called AMP945, now called namafotinib. We give that drug to patients. They take it as a pill in the days preceding their chemotherapy. And the chemotherapy they're getting in the ACCENT trial is gemcitabine and Abraxane. These two therapies that are really reasonably well tolerated by patients. So we started off that trial with a dose escalation phase that was trying to find a safe and well-tolerated dose, but that gave enough VAC inhibition so that we thought it'd be therapeutically beneficial. That was conducted a couple of years ago now. We identified a dose of 400 milligram dose, so that's not a huge dose for an oral drug. It's about normal for a lot of drugs of this type. So patients take that as a pill in the morning, once a day, and then three times in a 4-week cycle they will get the chemotherapy as well. And what we're looking for is showing that our combination of namafotinib and the gemcitabine Abraxane is better than historical data for gemcitabine Abraxane alone. So we did the dose escalation phase, identified 400 milligrams as a good dose to take forward. In the beginning of 2024, we then started recruiting patients. We recruited in total 55 patients. And that work was done both in Australia and in Korea. Korea, people always want to know, why did you choose to do the trial in Korea as well? Partly because Korea is has a very excellent medical system, great hospitals, great clinical trial capabilities. And they're an absolute pleasure to work with the people over there. But unfortunately, there is also a slightly higher incidence of pancreatic cancer in Korea as well. So we ran the trial in Australia and Korea, and it's just under 13 months, we recruited 55 patients. So we finished recruitment in mid-February this year. And what we've been looking at is monitoring the safety and the tolerability of the drug in combination with gemcitabine Abraxane, but also looking at the efficacy of that combination. And what we're seeing, if we look side by side with the historical data for gemcitabine Abraxane alone, is that our combination, adding on namafotinib on top of that chemotherapy, it is working better. So a couple of statistics just to illustrate that is that what's called the objective response rate. This is how many people are responding for 2 months or more to the drug. We're seeing a 31% objective response rate. That's confirmed. Confirmed meaning it's for more than 2 months, and that compares very favourably with the chemotherapy alone, where you're seeing only 23% of patients responding. So that's the first thing. If we look at progression-free survival, so how long are we keeping patients' disease in check, that it's not actually progressing so that they have to move on to a later therapy. Our figure, which we announced last month, was 7.6 months. So that's a full 2 months better than what's known for the chemotherapy itself, gemcitabine Abraxane. So it mightn't seem a lot, 2 months, but it's obviously very meaningful for patients. When you're talking normally to be sort of somewhere between 5 and 6 months, we're pushing that out now to 7 to 8 months. And the great news is, and what's not really apparent from that just single figure, is that we've got a subset of patients that are really doing very well on the drug. So we've got 7 patients who've been on the combination of the drug now for more than 12 months. 2 of those actually now beyond 18 months. So we're seeing a subset of patients having really prolonged activity and effect, keeping this disease under control. So that's really exciting. So we've got this efficacy, we've got this durability, particularly with some subset of patients. And then on top of that, across the board, we can confidently say that the addition of namafotinib to the chemotherapy alone is really not adding significant burden to the patients in terms of tolerability. So patients tend to have a lot of side effects from the pancreatic cancer itself, so vomiting, diarrhea, gastrointestinal problems in general. Then on top of that, they've got chemotherapy, which obviously has some effects as well, some fatigue and some nausea. So if we look at the figures for incidence of those side effects in our trial versus what's known historically, we don't see a big difference. So we do think the drug is not adding any burden to the patients, which is pretty exciting. And indeed, anecdotally, some of the clinicians we speak to say, look, in terms of tolerability, the drug's absolutely fine. It doesn't have any burden to the patients at all, which is really great to hear.
So how do we actually interpret it? How do we interpret the time that patients are on the trial? So I think it was like over 200 days median versus 117 in the benchmark study. How do we interpret that?
Yeah, the sort of figure for that, that's quite a simple calculation. I can do that calculation. Basically, we look at when patients start and when patients come off study. So patients come off study if their disease progresses, and generally that means the tumour's grown by more than 20% from where it started. They could come off because of other non-tumour issues. So they might withdraw consent, decide they've had enough. Often, most patients will come off because their disease has progressed. It could be pain, it could be the presence or the appearance of a new metastatic lesion, or it could be that the tumours are just growing. At some point, there's a resistance process that kicks in, or the tumour just hits another gear. When we're talking the median duration on trial, we're just really looking how long are we keeping patients so that they can stay on study, so that their disease is not getting to the point where they come off trial. So our data, you're right, when we did a data cut in July, it was 202 median duration on trial versus gemcitabine Abraxane, which is about 120 days. So already that looks much better. The progression-free survival calculation's a little more complicated because you're looking at what's happening to patients while they're on trial and indeed once they come off study as well. So it's a little bit more complicated to calculate that. And what we saw there is, as I said, a 7.6 months progression-free survival versus 5.5 months for gemcitabine Abraxane. And I need to stress, there's still 17 patients on study, so this data is still ongoing. We don't expect to have all patients off study for probably another 6 to 9 months maybe. And so these figures will evolve and will change as more data comes in. But at the moment, it's very promising that we're seeing a significant improvement on a number of these measures.
I guess there was the impression there were two well-publicised complete responses. One was written up in the papers and got a lot of attention, which is great. It would be great to talk about that. But I think also there was a perception in the market that maybe the median progression-free survival would have been better than that additional 2 months. So how do we square that circle? How do we make sense of the fact there's a small subset of patients that are doing really well with this 2-month increase? And then I guess the follow-up question is, how does that translate to overall survivability? Which presumably is the most important.
Yeah, just to be clear, the 2-month increase in progression-free survival is across all patients, not just the subset that are doing well. So there's a subset of patients who definitely haven't responded to the drug. They come off trial very quickly. We're trying to understand at a molecular level what's different about tumours for patients who do well versus those who don't do well on our drug. We don't have an answer to that at the moment, but that's obviously something we'd really like to know. But let me go back to the complete responses because I think they're important. That's very rare in metastatic pancreatic cancer to see a complete response, where basically all the tumours in the patient that you can visually see on the CT scans that are run every 2 months disappear. In other words, the patient, for all intents and purposes, is cancer-free. So that's really unusual. We saw one patient achieve that out of 55 patients in total. So already that's at a higher level of response than the historical data. We had this second patient that got a lot of publicity who was doing really well. There was still evidence of a small amount of cancer in their liver. And the medical team decided to actually go in and have a look. Is that really cancer or is it something else? And when they went in and cut out that tissue, it wasn't cancer at all. It appeared as if it was cancer in the scans, but it actually wasn't cancer. So that is known as a pathological complete response. So again, that's extremely rare. That one is extremely rare in pancreatic cancer that's metastasized. So both those findings are really good. And then we've got other patients who again have had very significant reductions in tumour size and are staying on trial for a long time. Across the board, we've had some patients who've come off study quickly, and we've had this subset, maybe 25% of patients who are showing this really long-term of benefit of the combination. We need to tease out why is that? What is it about their cancer that makes them respond better to our drug or to the combination of our drug and the chemotherapy. That's an ongoing process and we can't get hold of that. But as I say, the data's still early, Michael. It's interim data. We've still got a number of patients on study, and I expect that as these additional patients continue on study, that some of these figures will change as well.
Got it. But I guess even if it's effective in a small subset, that's still quite compelling, right?
Particularly if we can identify what it is, absolutely, because then you can actually assess patients prior to treatment and enrich the patient group for those who are more likely to respond. That's this process of sort of personalised medicine or biomarker-driven drug development, and we're very keen to get to that point. We're talking with a number of groups, both here in Australia and overseas, on how to tease apart the data sets we have and try and understand that. It's not simple, but I think there's something there, and we've got hints from some of the people we've been talking to that there is a sort of molecular signature that we could hone in on, but nothing to speak about yet.
Interesting. What's the plan with FOLFIRINOX then? I understand you're starting a trial. Is it the same design, but just using the different chemotherapy combination?
There's a couple of differences. So first of all, yes, it's the different chemotherapy. That chemotherapy is given once every 14 days, so twice a month. And the other difference is that because our drug appears to be well tolerated by patients, we'll be taking the drug every day. That's a little bit more convenient for patients. They don't have to know, do I take my drug now? When am I getting my chemotherapy? I start taking my drug, which is what they currently do with the Accent study. So going forward, because the drug's been well tolerated, because we believe it has this activity on its own, it doesn't just need it doesn't only need the chemotherapy to work. That we're going to daily dosing of nalmefotinib and combined with FOLFIRINOX. So that's a trial that we've opened up here in Australia. So we've got 2 sites, one in Sydney, one in Melbourne, that are recruiting patients already, and we've announced that we've put our first patient on study, which is great. We've got 4 sites identified in the US. I would have liked to be telling you that they're already recruiting patients. There's been a little bit of a delay in actually getting some final. It's a purely paperwork at each individual site has their own special processes we have to go through. So it's been a little bit slower to get the US sites on board, but we'll get them on board very quickly in a matter of weeks so we can start recruiting patients there. This is a trial that's been run under investigational new drug application from the US FDA. So they've reviewed the protocol, they've reviewed the safety, they've reviewed everything about the drug product and are comfortable with that package we sent to them. So that trial is now underway. We're starting with a dose escalation as we did in Accent. We expect a dose escalation to be complete in Q1 of next year. And it is a different chemotherapy. It's more aggressive, as I said earlier on. How that's going to impact the safety and tolerability of the combination with nalmefotinib needs to be determined. We don't know yet, but everything we've seen to date makes me think that combination will be tolerated. And from our preclinical studies, we're very confident that adding nalmefotinib on top of FOLFIRINOX will improve the outcomes. So we now have to show that in patients.
Sure. So when we think about timeline, we've still got those existing patients on the Accent trial, the FOLFIRINOX trial. What's next?
The Accent trial should probably finish, as I said, probably sometime midway through 2026. Because we've got this very promising data to date, the anecdotal feedback from the investigators has been very positive as well. We are now planning a registration-enabling study of nalmefotinib in combination with gemcitabine and Abraxane. That planning takes a long time, so we're starting that process now. We'll be going to the FDA very shortly with a series of questions about our proposed trial design and just seeking their advice on whether our approach is appropriate. If it is, we feel we've got a pretty good case to make based on the data we have to date. That will then write a full protocol and start engaging with potential clinical partners, other groups to help us run that trial in the US, but also in Australia, Korea. We'll continue with Korea and then in other parts of the world as well. So that's the sort of plan. What we would hope, if the engagement with the FDA goes well, we do have a fast track designation with the FDA. So we've got it's like boarding an aircraft. We're in the kind of priority lane already. That will get good feedback from the FDA. Obviously, timelines at the FDA have been impacted by a number of people leaving that organisation, but we hope we'll have good interaction from the FDA over the next few months and then into the new year, so that we could, in theory, start that registration study at the end of 2026. That would be a really good timeline if we can get to that. Meanwhile, of course, we're running the Amplicity trial, so this is the combination with FOLFIRINOX, and we'll be getting data from that study as well. We feel it's really important to have demonstrated activity with the two main chemotherapies that are used in pancreatic cancer, because, as you said earlier on, Michael, FOLFIRINOX seems to be preferred in the US, so we want to show that our drug can work with FOLFIRINOX. We want to show that it can work with gemcitabine Abraxane. Because we started with the Abraxane, that will be the one we'll take into a registration pivotal study sooner. And then we'll have to make a decision how we move forward with the FOLFIRINOX combination work. So that's going to be dependent a little bit on the data and how things are working with the Abraxane combination.
There have been some positive results from other approaches in pancreatic cancer, like KRAS. How do you see those fitting in? And is there an opportunity there if your drug is helping activate these tumours or turn them from cold to hot? You think about other combinations, other potential partnerships that you could pursue?
Absolutely, yeah. So this whole field, I think, for a long time, pancreatic cancer was seen as a bit of a very challenging place to do drug development. And our data, and there's a few other approaches that they're gaining a bit of traction. So one of them is KRAS inhibitors. These are a new class of drugs that target an oncogene that's present in, and it's mutated in a lot of pancreatic cancer patients. The data from the trials to date has been positive. KRAS inhibitors do work in pancreatic cancer patients to an extent. Their resistance does develop, unfortunately, and they do have some reasonably challenging side effects as well. So the sort of lead asset is being developed now in the second line patients, so patients who progress from chemotherapy. And the data there's promising, I think. So I think KRAS inhibitors will probably end up as a part of the treatment regime for pancreatic cancer patients where the disease is metastasized, initially as second line. Will they make a headway in first line therapy? I think the jury's out still. We just need to see what is the toxicity profile, what's the response rate in those patients. What I can tell you is that our own data and the data that's in the scientific literature shows is if you combine a FAK inhibitor with a KRAS inhibitor, you get a better response. You're breaking down that fibrosis, so more KRAS inhibitor can get into the tumour. But also you're blocking a resistance pathway that often develops in response to KRAS inhibition. So you've again got this double whammy of an effect on the microenvironment and effect on the cancer cells themselves. So we think there's a real opportunity of combining a FAK inhibitor and a KRAS inhibitor as well. And we're in early discussions with some of the KRAS inhibitor developers about looking at some combination studies to see if we can enhance the activity, improve the efficacy, and possibly allow those drugs to be used at a slightly lower dose so that you're dealing with some of the toxicity and down-tuning that.
How does that work in practice? Like, you'd need to partner with another company to run the trial together, or would they run the trial and you provide the drug? Like, how?
There's a number of different models. One, it could be that we run the trial and just have an agreement to get access to their drug. Another would be we go in together and we each pay 50-50 kind of thing. These are discussions that are ongoing. We've recently completed a fundraise of $27.5 million Australian, which is ostensibly to continue to support the Accent and Amplicity trials and start planning for this registration study. But there is a small amount of money put aside in there to support investigator-initiated studies in pancreatic cancer in combination with KRAS inhibitors. We're in discussion with now some investigators and through that in discussion with some companies developing KRAS inhibitors. So hopefully one of those discussions will be successful, and we can test that combination and see if we do add value, do add a benefit to KRAS inhibition, and make those drugs work better, and creates another avenue where narmofotinib can be used on top of improving outcomes with chemotherapy. It can be used then in combination with KRAS inhibitors. I did want to mention another area that's worth noting in terms of pancreatic cancer, and that is antibody drug conjugates. So these are biological agents, so they're antibodies that have been armed with a payload of a very toxic species. And the idea is that the antibody finds a particular antigen on the cancer and then delivers that toxic payload to the cancer. So it's, if you like, directed chemotherapy, really. But again, an antibody can't get to the cancer cell if there's a fibrous shield around it. We think there's really exciting opportunities for combining narmofotinib with the antibody drug conjugates that are in development in pancreatic cancer as well. So that's another area. We're talking to other people about that. And again, really what I'm trying to position here is that there's a really long-term opportunity as pancreatic cancer treatments develop and new ones come online. Narmofotinib could actually be almost like a maintenance therapy that just sits with patients throughout their journey, improving the efficacy of each of these therapies along the way. So that's sort of my dream, if you like.
Yeah. And so how does it work? If you're being used in combination, just what... Are you able to comment on the commercials, how much you could charge, how big these markets are?
Yeah, so
Particularly if it's used in combination.
So the combination pricing can be a little bit complicated, but at the moment, I think with both FOLFIRINOX and gemcitabine Abraxane, they're essentially generic. Abraxane's coming off patent now, gemcitabine's off patent, and FOLFIRINOX is off patent. So those therapies are really quite cheap, and then we'll be adding our drug on top of that. Our drug's a small molecule kinase inhibitor, and the pricing, at least in the US market, is somewhere sort of between 120,000 a year to double that, 240, 250,000 a year. So we've done a little bit of pricing assessment, and we think we'll be able to charge in that kind of window on the US market. Co-development with an asset like a KRAS inhibitor that's still on patent and still therefore charged at a higher rate, that's something that we'd have to work through with the developer of that other asset. But we do think, based on what the market is looking at in other areas, that there's good precedence for us to be able to charge and to be reimbursed at very good pricing.
And who's currently marketing those chemotherapy agents?
So the chemotherapies, gemcitabine was originally marketed by Lilly as Gemzar. Abraxane, I think, is with Bristol Myers Squibb, but there's generic versions that are coming online. And then we've got FOLFIRINOX. There's just a range of generic players in that space. So in terms of innovator companies, a lot of companies are getting into the pancreatic cancer space because it is, an untapped market really, and the increasing prevalence means that it becomes, something that I think people are seeing there's a commercial opportunity there with the right asset.
Got it. And in terms of the next set of trial results, maybe the end of Accent and Amplicity, what kind of results do you need on survivability to get the market really excited and payers excited and clinicians excited?
That's a great question. Because metastatic pancreatic cancer is really so devastating, you don't need a lot for people to be excited. So the last drug to be approved in metastatic pancreatic cancer was a drug called Nalirifox, so it's similar to FOLFIRINOX. That was approved in 2023 by the US FDA, and that saw only a 2-month improvement over gemcitabine Abraxane. That was considered not only enough to actually warrant approval, it was also met with a lot of enthusiasm by the patient advocacy groups, by clinicians as well. Anything above a 2-month overall survival improvement would be considered significant. We'd obviously, the bigger that window, the better. We'd obviously hope that we'd see something more than 2 months, but even at 2 months, I think that'd be good in terms of an overall survival figure. put that hand in hand with the fact that, the drug's well tolerated. I think that sort of sweetens that figure as well, because we're not adding that toxicity burden. So yes, we've got that. If we can identify a biomarker or biomarkers that actually identify patient responders even more, then indeed we'd be looking at seeing an even better improvement, I'd suggest. But all that work is, going on in the background. Nothing to report yet.
Got it. So why don't we just take a high-level view of like kind of the mid and long-term plan here? So you've got to finish this current trial, new trial with FOLFIRINOX, hopefully a registrational trial, and presumably that will require either a partner or funding. Are you able to just take us through the strategy over the midterm, the next few years?
So look, the near term is for us to obviously complete the ACCENT trial, report on the overall survival data. We'll probably report on that, at least have a pretty good number by Q1 next year, and then start planning what the registration study looks like. We're going to get some pricing for that study. We've got some rough guidelines now, but we want to, once we've had this engagement with the FDA and we've got a clear idea exactly the sort of patient numbers and the approach that we can adopt, we will speak to the clinical research groups and start pricing out what that trial looks like. It is possible, based on the quality of the data we've got, that we could find ourselves in partnership with a pharma partner to help us run that clinical study. But if not, then we'll be looking at ways to fund that work through money we have in the bank at the moment, and through obviously an additional raise later next year. So that's or into 2027, depending how our funding goes. And we are looking at some regional licensing deals, so that's something we're very keen on. It's obviously a model that other Australian biotechs have used well, to have a regional partnership that obviously brings in upfront money and then milestones and royalties. So that's something we're actively pursuing. I think there's some good opportunities. We're obviously doing our study in Korea at the moment, so there's good opportunities there, but also in China as well. So we're looking at that. So that's the near and medium term. I think longer term, as we think about progressing the compound into a registration enabling study, we're a small biotech in Australia. We're not going to be the biotech that's going to be out there marketing this drug. So we will be looking for partners during the process of running a registration study. We'll be obviously looking at the data at the end of this current trial that's coming out of the Amplia trial, using that to build relationships with pharma partners. So we'd be looking to identify a pharma partner that we could either co-develop or license the drug to down the line. So that's the model, and so far the data's been good, that there's interest from the BD front, from pharma and biotech, both worldwide and in the region. We'll see where that pans out.
Got it. Is there anything that we've left out, anything we've missed, any key points that we should cover?
I think it's been a very extensive discussion, to be honest. I hope I haven't gone into too much detail along the way, but no, I think we've covered everything that we needed to, Michael. That's very much on our radar. The opportunities, that we see for the molecule and the program are really significant, and the data to date is just backing up our confidence that this is a really good drug, safe, well tolerated by patients, and does appear to be enhancing the activity of chemotherapy. We've not spoken about other opportunities for namafotinib. We set out thinking that pancreatic cancer would be just one of a number of fibrotic tumours we'd look at. One of our competitors is a US company called Veristem. They've recently had their drug approved in low-grade serous ovarian cancer. Their drug, it's as a combination therapy. We get a lot of interest from ovarian cancer specialists who are interested in the opportunity of namafotinib in ovarian cancer. So that's something we're looking at. I think we have to, obviously, we don't want to spread ourselves too thin. We have to be careful and mindful of spending our money appropriately on the trials that we've got going. But, if there's an opportunity to explore namafotinib in an ovarian cancer indication through, say, funding from a charity or grant, then I think we'd jump at that chance, because there is data that we've got. There's data in the literature that says a FAK inhibitor in ovarian cancer has real potential. So that's something we'll be looking at as well. So that's something we didn't speak about, but something that's happening in the background as well. And should there be any developments, we'll obviously announce that to the market.
Okay, great to know. Well, why don't we wrap up there? Thanks so much for coming on.
Thank you, Michael. Appreciate the chance to talk, and look forward to catching up again. Cheers.
Absolutely. Thanks
All the
again.
best. Bye-bye.